Hey guys, so welcome back to our bioinformatics lectures. Obviously, this is much different than an in-class lecture, so this is the best we can do with our lectures in an online format. And I know it's been a little bit, so we had spring break 2 weeks ago, and then we had another week to kind of prepare for this online format. And so this week is something I'm sort of treating as sort of a reset week, so we can sort of remember where we left off and sort of jog our memories. So all of this material that I'm going to share with you today um, is going to be from previous lectures. So if you ever have, a question or something's a bit confusing because I, kind of share big ideas from a lot of the previous lectures, make sure and go back to those individual lectures themselves and see if you can answer your question of confusion. If not, then feel free to ask a question on the discussion board or through the Remind app, whatever is the easiest for you to contact me. Okay, so, and also I did want to remind you, with this sort of reset week, if you haven't turned in, any of the in-class activities or assignments, now is a good time to kind of, evaluate all that's due and what you haven't turned in and get those things in. All right. So this is our semi-quick review. And remember that now we are, at least the last few weeks before the spring break, we were working through this example NGS pipeline, and we were trying to follow it as closely as possible. So remember, before that, we had done a lot with, looking at and evaluating these FASTQ files, the large text files that have a lot of our short sequencing information that we get from the next generation sequencer. And now, not now, but previously, we had done some quality control on those using a program called FastQC on the Galaxy server. And now we're sort of taking those FastQ files or short sequencing reads, aligning them to the genome. We want to clean up that alignment. We want to perform what's called variant calling, and then we want to annotate those variants, and then hopefully, we have a goal of identifying a specific genetic variant that's important. In our case, with our activities, it happens to be in health and disease. And so remember way back that essentially we are chopping up this extremely long genome of about 3.2 billion base pairs, and we are creating these very short sequencing reads that come off of our NGS sequencer that we can see there. And so we kind of broke this down in the weeks preceding spring break. There are two major ways to read the genome essentially in a next generation fashion and analyze genetic information. And I think they both have their merits. We're going to go through both of them. Right now we are focusing, I'm going to get my laser pointer out. We're focusing entirely on this right side of our little figure here. Next week we will go through genotyping and what's called genome-wide association studies. And so now what we're trying to do is we're performing what's called whole exome sequencing, which is very important to detect specific variants that are very rare in the general population. They have a very large effect, meaning that the percentage of people that have that particular genetic change will largely have, disease X or disease Y or whatever phenotype or biological trait we happen to be interested in. These are normally termed Mendelian disorders because they follow a very characteristic inheritance pattern, where if the person inherits, one copy of that defective variant, they tend to have that disorder. And so we're looking at whole exome data, or we were before spring break and all of the kind of pandemonium hit. And so you may be asking, why would we focus on the exome or all of our exons? So remember, the exome in this case being all of the exons of the human genome, and exons being what are immediately translated into proteins. And the answer, from a very basic sense, is that we understand a lot of, how these exons are translated into proteins, and how changes in that amino acid composition of that protein can affect function. And so it's the part that we actually know best of the genome. And also kind of a cost consideration and a time consideration, breaking it down to the exome as opposed to looking at the entire genome, right? We are cutting down our search area by orders of magnitude. And so one of the deliverables from the Human Genome Project was that we found that only about maybe 1.5% of our genome was actually exon based and actually immediately translated to protein. And so taking out that 99% that we don't need to worry about drastically cuts down our search area from 3.2 billion that you can see there on the screen to about 30 million base pairs. But you can see from a computational perspective that nothing really changes for us, right? So the NGS instrument still delivers FASTQ files. We're still manipulating those FASTQ files, which is the large text-based file that's just a huge listing of all of our short sequences. We're going to align those reads to the reference that was developed during the Human Genome Project. We're going to basically have what's called variant caller program tell us where our sequences in our FASTQ files differ from that reference, and then we're going to characterize those genetic variants somehow and try to determine where the disease causing variation actually is in the genome. And so here's kind of just a rundown schematic of what we've previously covered. So remember, we had a question on, I believe it was Quest 2, about doing some sort of what's called de novo assembly with these short reads, right? So finding that microhomology, or that small overlap between sequences to kind of build that consensus. And now we're moving to more of a mapping approach, where we take those billions of short reads and we just align them to a reference, and then perform some other types of analyses to tell us where our sample DNA is different from reference. And so remember that all of our sequencing-based aligners are going to align to our human reference in this case, or whatever our model organism tends to be. So this can be mouse in a lot of research. This can be yeast. This can be any other type of model organism with that has had the genome sequence. Remember, all of these sequencing aligners kind of perform the basic function of aligning these sequencing reads, and they need a way to be able to actually score those alignments and actually output the best alignment for that particular short read. And so this generally breaks down to some sort of reward for a type of match between the reference and the sample DNA, and then a penalty, obviously, for mismatches. There's a penalty for gaps that are created, and there's a reward for closing particular gaps. And so it's important to mention that different tools that we use, so there are a number of different sequencing aligners out there, they use different scoring values and give different results. And so we need to really know our data to determine, what's the best sequencing aligner to use in our particular case. Now, this, we would probably think, just intuitively, this would be pretty easy if we were doing it, like one read to the reference. But, as we can imagine, there are a number of different ways that we can align a short sequencing read to reference. And, you can see there, with our reference and our observed sequence in our short sequencing reads, that we have 4 different alignments, and dependent on the particular tool that we use, we will get, different scores, and it will evaluate, which is the best alignment. And in fact, these are all different productions of, the alignment of that particular sequencing read to the reference using different tools. And so you can see that, using different tools will give you different results. And so we need to keep this in mind, as we choose a sequencing aligner to use for our purposes. And obviously, the tool that we use will impact our downstream analysis, because the tool that we use, how it aligns that short sequencing read to the reference, will ultimately determine what variants are called in which position. So you can see all of the different sequencing aligner tools that we just said, are published in the literature. They're well vetted. They've been used a lot of times. They all give us different results, and it's with the same sequencing read. And so you may have remembered that, we played a little game, and, you went to this website and you kind of practiced doing sequence alignment yourself. Hopefully you saw that this wasn't as easy as just like, oh, there's just a perfect match every single time, and you had to kind of move around, create gaps, close gaps, and see, the best score that you can possibly get. And then I had you actually do the 3 levels that you see there on the screen and output a screenshot of you solving those 3 levels as an activity. So if you haven't done that, if you haven't turned that in, please go back to that specific lecture and get it in. And next, we will continue with the variant calling portion of our review. All right, see you in a bit, guys.